Lecture 8. Renal and circulatory effects of aldosterone. In this lecture, we'll continue with the effects of aldosterone. Excess aldosterone causes hypokalemia and muscle weakness because it causes the secretion of potassium into the renal tubules and also the entry of potassium into the cells. So that these effects causes hypokalemia, which is a decreased potassium concentration. And because of this decreased potassium in the extracellular fluid, muscle weakness develops. Now this muscle weakness is caused by a change in the electrical potentials of the nerve and muscle fiber membranes, preventing transmission of normal action potentials. Now this increased tubular hydrogen secretion into the renal tubules in exchange for sodium reabsorption in the principal cells of the renal collecting tubules. This leads to alkalosis. Now, on the other hand, less aldosterone causes hyperkalemia, which is an increase in the extracellular fluid potassium concentration. And when this concentration rises more than 50% above normal, this can lead to cardiac toxicity and development of arrhythmias. Effects on the sweat and salivary glands. Aldosterone stimulates sodium and potassium transport in the sweat glands. Now this effect is of great importance during acclimatization in the hot weather. So aldosterone causes the transport back into the ducts of the sweat glands to preserve the sodium in the extracellular fluids. Now similarly, it stimulates the sodium and potassium transport in salivary glands as well as in the intestinal epithelial cells, especially in the colon. Now this prevents loss of sodium in the stools. Otherwise, if this sodium reabsorption is poor, this will lead to failure to absorb chlorides and water as well, leading to diarrhea. Coming to the mechanism of action. Now, because of its lipid solubility, aldosterone can diffuse readily into the interior of the tubular epithelial cell. Once it enters the cytoplasm, it binds with its receptor protein. Now this aldosterone receptor complex diffuses into the nucleus, where it induces some specific portions of the DNA to form messenger RNA. Now this messenger RNA enters the cytoplasm and causes the formation of proteins. Now these proteins can be sodium potassium pump, which helps in pumping the sodium out of the principal cells into the renal interstitial fluid, thus causing low sodium gradient in the cell. And it also causes the formation of protein channels, which are known as epithelial sodium channels. Now once these channels are inserted, they provide channels for the sodium to move into the interior of the principal cells down its concentration gradient, which is created by this sodium potassium pump. So this is the mechanism of action of aldosterone on the principal cells in the kidney. Coming to the regulation of aldosterone secretion, there are four main factors which regulate aldosterone concentration in the extracellular fluid or in plasma. These include potassium ion concentration, angiotensin II concentration, and these two are the main factors which increase the aldosterone concentration in the body fluids. The other factors include sodium ion concentration and ACTH from the anterior pituitary gland. Now let's study the adrenocorticotropic hormone before we move on to the study of the glucocorticoids. The adrenocorticotropic hormone, or the ACTH, is secreted by the corticotropes of anterior pituitary gland in response to corticotropin releasing factor, which is a factor secreted by the neurons located in the paraventricular nucleus of the hypothalamus. Now, ACTH is a part of a bigger molecule called pro-opiomelanocortin. So pro-opiomelanocortin is basically a pre-pro hormone. So first there is formation of a pre-pro hormone called pro-opiomelanocortin, POMC. Now this bigger hormone, bigger molecule, POMC, is the precursor of many other hormones, of which ACTH is one. So ACTH and melanocyte stimulating hormone, beta lipotropin and beta endorphin, these are all derived from this bigger hormone, a bigger molecule called pro-opiomelanocortin. Now, the precise type of products formed from this POMC or pro-opiomelanocortin depends on the types of the processing enzymes present in that specific tissue. And these enzymes are called prohormone convertases. For example, if ACTH is to be formed in the anterior pituitary gland cells, then it depends upon which type of the processing enzyme is present in the anterior pituitary gland tissue. So at the top you can see the pro-opiomelanocortin preprohormone molecule, and these red and blue arrows show that these are the prohormone convertases. So they act or slice the parent molecule, the bigger molecule, into many products like ACTH, beta lipotropin, and which is further divided or sliced by this PC2 into gamma lipotropin and beta endorphin. Now, the mechanism of action of ACTH. The ACTH binds to receptors on the adrenocortical cells in the zona fasciculata and zona reticularis. Now, this leads to the activation of adenylyl cyclase enzyme, causing an increase in the cyclic AMP, which activates the protein kinase A enzyme. Now, this leads to the phosphorylation of an enzyme called cholesterol ester hydrolase. Cholesterol ester hydrolase. Now, this is present in the it causes the conversion of cholesterol esters and changes them into cholesterol. Now, this cholesterol enters the mitochondria, where it undergoes many intermediary steps to form the cortisol. Now, cortisol is the major glucocorticoid. ACTH is secreted in irregular bursts throughout the day. It has a circadian rhythm. Now, in humans, the bursts are more frequent in the early morning, and they are less frequent in the evening. Now, having studied the ACTH, which acts on the adrenal cortical cells to synthesize the glucocorticoids, let's now study the glucocorticoids, which are the second major type of hormones produced by the adrenal cortex. The glucocorticoids include cortisol, which is the major glucocorticoid, and then there are other synthetic glucocorticoids also which are used in clinical practice. These include the prednisone, methylprednisone, and dexamethasone. The cortisol. Cortisol occurs in the plasma in three forms: as free cortisol, and protein bound, and as its metabolites. Mechanism of action of cortisol. Cortisol interacts with intracellular receptors in the target cells. Inside the cell, cortisol binds with its protein receptor in the cytoplasm. And this cortisol receptor complex then enters the nucleus of the cell, where it interacts with its glucocorticoid response elements. And this interaction with glucocorticoid response elements induces or represses the gene transcription. The regulation of cortisol obeys the negative feedback mechanisms. So here you can see in this figure that the hypothalamus is excited by conditions of stress, which may include trauma, infection, intense heat or cold, surgery, or any debilitating diseases. So these types of stress excites the hypothalamic neurons to secrete the corticotropin releasing factor into the median eminence, where through the portal vessels it reaches the anterior pituitary gland and causes the release of ACTH. Now, ACTH acts on the adrenal cortex, where it leads to the synthesis of cortisol. Now, cortisol has its effects on the glucose, protein, and fat, and other effects that we are going to study shortly. And these effects then causes the relieves the stress. And in addition, the cortisol also inhibits the release of ACTH and the release of corticotropin releasing factor from the hypothalamus. So the regulation of cortisol is by the negative feedback mechanism. Now, similar to the circadian rhythm of the ACTH, the cortisol also follows a circadian rhythm. So the secretory rates of CRF, ACTH, and cortisol, all of them, they are high in the early morning but low during the evening. Now, this results from a 24-hour cyclic pattern in the signals coming from the hypothalamus. Now let's study the effects or the actions of cortisol, which is the major glucocorticoids. So we can say that these are the effects of the glucocorticoids or steroids, as they are commonly known to the layman. The effects on the protein metabolism include the increased catabolism of proteins, leading to gluconeogenesis. And you know that gluconeogenesis is the formation of glucose from the proteins, and this leads to a negative nitrogen balance and causes the hyperglycemia. Effects on the lipid metabolism. It causes an increase in the cellular lipase activity, which causes the mobilization of fatty acids in the plasma. Therefore, there is hypertriglyceridemia produced by the cortisol, high cortisol levels, and this may lead to the myocardial infarction. On the carbohydrates, it has an anti-insulin effect, and therefore reducing the carbohydrate utilization in the peripheral tissues, and this increased glucose in the blood leads to hyperglycemia and also glucosuria, which is the spilling over of glucose in the urine. So there is more glucose in the urine because of hyperglycemia. It causes osteoporosis because the cortisol or the glucocorticoids, they decrease osteoblasts, and they have an anti-vitamin D action. That is, they cause a decrease in the absorption of vitamin D. Effect on the blood cells. The cortisol or the glucocorticoids stimulate the erythropoiesis, and therefore they increase the neutrophils and the platelets. But there is a decrease in eosinophils, basophils, and monocytes. The effect on the lymphoid tissue is that there is a fall in lymphocyte count, and they inhibit the formation of interleukin 2. On the GIT, they may cause peptic ulcers by causing an increase in the gastric acidity and the pepsin production. And they also cause thinning of the gastric mucous membrane. Then very important action is the anti-inflammatory action. Now this is because that the glucocorticoids or the cortisol stabilize the lysosomal membranes in the cells. So these stabilization of the lysosomal membranes leads to a decrease in the release of these lysosomal enzymes, and this causes a decrease in the inflammation. And similarly, they decrease the capillary permeability, and they also cause a decrease in the migration of the leukocytes to the site of the inflammation. Now in the end, And there is a question and list factors known to play roles in the regulation of aldosterone. MCQ. ACTH is increased during periods of stress or decreased during period of stress, inhibited by aldosterone or stimulated by glucocorticoids or the epinephrine. MCQ number 2. Anti-inflammatory effect of cortisol is due to increased of which of the following options?